This is TWIS. This Week in Science, episode number 803, recorded on Wednesday, December 9th, 2020. Bringing light to the darkest timeline. Hey everyone, I'm Dr. Kiki, and today we will fill your head with issues, poo, and beavers. But first, thanks to the Burroughs Welcome Fund and our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. Of everything that has ever happened, ever been done, or occurred in the world, this is the end. The end of everything past. This is the result of the experiment. This is now. And like all nows, we are at the beginning. We are at the designing point of the next experiment. We are at the birth of the future. This is the prelude to everything that is yet to come. The moment of now comes with great responsibility. Choices now will affect nows to come. And now is the only moment in which you can do anything about it. If we make choices based on science and reason and empathy, we can build a future brighter than the one we started with. If we ignore science, if we act without reason, without empathy, we will fall into a future far worse than where we started. Whichever world we end up with, you have at least made one choice that can only help improve our chances. You are now listening to This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back. We're here. We're ready to talk about science. Ooh, bring in the science light. That's right. Because you know what? Knowledge, it is a light in the dark. And we're here to do that. Everything's yet to come, and we can hold that candle to the darkness. I have stories tonight about humans and nature, some lightness, some supremacy, and brains, because I like them.
Brain supremacy.
Brains. So yes, right. Fully brain supremacy. That's totally what it is. Justin, what did you bring?
I've got cat scratch neuropsychiatric issues, call to action, a new Neanderthal cemetery, and do police really need to look and act like the military?
The answer is no, but I would like to hear the story later.
No. But there's a study that has been done along these lines.
Great. Blair, what's in the animal corner?
Oh, I have beavers. I have animals with COVID. I have dog brains, and I have pandas covered in poop.
There's the poo. We knew it would be there eventually. I'm sure this story just is one more reason to love pandas.
Oh my God, I cannot wait. I don't want to hype it up too much, but I feel like this might be my favorite story of 2020. So bear with us here. Get it?
Or
bear with us.
Panda with us.
Is it a bear story? Okay. Yeah,
Panda. All right, let's dig into this science. But before we do, I would like to remind each and every one of you that if you are not yet subscribed to This Week in Science, you can find us all over the place. We are a podcast. You can subscribe to us as a podcast, download us to your device, look for This Week in Science in every podcast directory. You can also find us on Facebook, YouTube, and Twitch. Look for This Week in Science, or on Twitch, it's TWIS Science. You can visit twis.org for information. Okay. Time for the science. The unbearable lightness of quantum supremacy. That's what I want to start with.
Okay.
Yeah. All right. There has been a race to build quantum computers. There have been a couple of different designs for quantum computers so far, and they're big and they're bulky and they have reliability issues. And so far, they could potentially, as they grow, be amazing for servers and services that are large scale, where you have a building to hold your quantum computing center in. But there is another alternative design that consists of optical light, laser beams, because, well, not just because lasers are awesome, but because you can control light. You can use crystals and mirrors to bounce it different directions, control its polarization, to really control the way that it works. And last year, a Chinese quantum computing engineering team, after Google announced that they had reached quantum supremacy, which is still debatable, there was a Chinese team that announced they had created a computing device that could potentially also have reached quantum supremacy. They just published about this device of theirs. It is a really amazing design. The design uses lasers and beam splitters. And that's a fairly simple design, but it has to be very, very, very controlled because you're dealing with the detection of photons and the detection of not just any photons, but identical photons, figuring out which way they've gone as they have hit the beam splitter and diverged along their path. Now, the thing about this quantum computer is that it was built to do this problem of detecting and figuring out the probability of where these identical photons would end up once they went through their beam split paths. It solved the problem in under 4 minutes, whereas a classical computer, like the ones that we're running the show on right now, would take about 2.5 million years to calculate the same problem.
Okay, that I was gonna say you're gonna like that. My computer even could do that. Because I was like,
it would take a very, 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 very long time.
How long does it take to do the calculation to figure out that it would take 2.5 million years?
Oh, that's a very good question. Yes. Probably a napkin, back of the napkin bar table calculation, I'm sure, with these engineers. I'm absolutely sure. The classical computers, no, it wouldn't be your desktop computer. We're talking about probably supercomputers. It would take the most powerful computer, classically designed computer with circuit boards and everything, 2.5 million years to calculate this problem that was solved in under 4 minutes by this optically based quantum computer. This is amazing. It's a huge breakthrough, but this computer was only designed to do this one problem. You can't program it. It's not going to be able to do any other kinds of problems. So is it really a computer? Not really. It's just it's a calculating device, but just a very
like a function machine, like
a specific function machine. Yeah. Right. Yeah. Yeah. So very exciting development because it could lay the groundwork for future optical quantum computers that, based on their design, would take up less space, less energy, could potentially be more useful generally than other designs that are out there.
Quick question about this. Not crazy quick, but quick.
Quantum quick.
Do they have an estimate of how long it will take before the experiment starts? Because I just imagine that's a very long 4 minutes. So it's like, 4 minutes is it's very short for something like this. But in terms of it taking a computer doing a thing, nowadays 4 minutes is a really long time, right? So you can imagine kind of the researchers kind of sitting there for 4 minutes. You're like, is it broken? Is it going to work? Is it going to take 10 years? Right. Is it going to take 5 minutes? How long is it going to take? They must have had like a rough
No, idea, maybe not, right? Though. Maybe not. Because maybe this is the great thing. They spend, 2 years getting this thing built, overthinking everything, designing it.
Yes.
They get to
the one
now
thing.
We're running. All right. Okay, I guess we're gonna take shifts. We have this elaborate alert system to let us know if anything happens. Mostly, we just want to make sure the power doesn't go off in case there's a storm next winter, You know, got the air conditioners, all coolers are all ready for this, for the heat in the summer to keep all the servers going. Wait, what? We're done? Oh. Did we even buy champagne to celebrate? I don't think we even had time. That's good. Yeah, I think it probably happened amazingly quick, like compared to like all the time that went into setting it up.
Yeah, the time setting it up, and there's a fun comment in one of the articles. Chris Lee from Ars Technica has written a great article about it. There's a comment he makes about there are some very proud graduate students out there for the work that they have done to make this happen. Yes, to bring it together. So the beam splitters and the way they split the photons, 2 photons arrive at the beam splitter at the exact same moment, and they have a chip that has the equivalent of 300 beam splitters. They have 50 inputs, 300 beam splitters, and the possible output states the number that are available is about 10 to the 30. This is a huge, huge possibility of states. So the photons get sent in, and then they exit in this random way from all the possible states. The calculation of that, they do it over and over and over again, but the calculation is much faster. Justin, are you going to tell me about cats right now, or are we going to talk about a disease?
Oh, no. This is an infectious disease story. There is it's an emerging infectious disease that threatens humans. Yes, it's not the COVID-19, which, while on the rise currently, hopefully will be declining once we start vaccinating people and, going outside after not going outside for a while still. Gotta still stay home and mask, all that kind of stuff. Sciencey magic is on the way, though. Meanwhile, there's a bacteria which is known to cause fever and rash that has also been now recently found to cause skin lesions and neuropsychiatric symptoms of mental illness. This is a study by North Carolina State University researchers. They found additional instances of Bartonella infection in humans who exhibited these neuropsychiatric symptoms. This is adding to a body of evidence that not only can this infection mimic a spectrum of chronic illnesses, including mental illness, but also that dermatological symptoms may accompany the infection. So this is a bacterium historically associated with cat scratches.
Cat scratch fever? Is that what you're talking about?
This is cat scratch fever.
Yeah.
Which it's thought to be short-lived, have a very limiting infection time period, but it turns out that's probably not as true as we thought it was. There's actually 30 different species of this, 13 of which have been found to infect humans, and so they're working on lots of ways of detecting it. It's notorious for hiding in the lining of blood vessels in the skin. But yeah, so this is building off of previous research. This is Edward Breitschwood, published a case study involving an adolescent boy who had been diagnosed with a rapid onset of schizophrenia. This is not something that, rapid onset means it wasn't there, and then next week, now he's exhibiting all these schizophrenic signs. He also happened to have these skin lesions. So the research group documented Bartonella infection, and the patient received antimicrobial therapy. All the neuropsychiatric effects and symptoms resolved themselves. Boom. Fixed it. Fantastic. So this is the follow-up study from that work, published currently in the journal Pathogens, that 33 participants suffering from neuropsychiatric symptoms. This is everything from sleep disorders, migraines, depression, anxiety. 29 of the 33 participants were found to have Bartonella infections.
Huh.
Yeah, based on the blood work that they did, PCR testing of the serology samples. 24 of the 29 positive participants, that's 83%, reported the appearance of skin lesions during their illnesses as well. So there was also accompanied, and by skin lesions, there's, depending on what you've got, it can be kind of different, but it's sort of the one that they were seeing here was kind of interesting. It was basically stretch marks.
Yeah.
They were getting stretch marks, and these were, they of course ruled out things like bodybuilding or rapid weight gain, loss, pregnancy, all those sorts of things that you might normally get stretch marks from. These people suddenly had stretch marks and sort of in weird places too, maybe that weren't normally associated with where you would get stretch marks, but these sort of reddish, long, elongated lesions. There's other ones too, but I'm not going to talk about those. But yeah, it turned out like a lot of these cases were just cat scratch fever. And this is notoriously affects children predominantly. They tend to get this more than adults. Apparently, if you go to the CDC's website on this, there's a really cute picture of a kitten.
I'm looking at it right now. It's very funny.
So apparently kittens transmit this even more so than adult cats.
Oh, that's interesting.
And another vector
they don't clean themselves as well.
I don't know why it is, but but another vector is fleas. However, they're the transmission, one of the transmission sources, but the vector really is cats. Cats are the ones that are harboring this bacteria. So if Toxoplasma gondii wasn't enough, maybe cat scratch fever will help convince you. But isn't it weird that everything we're getting from cats causes psychosis of some sorts? Schizophrenic side effects? Maybe that just explains why cats are the way they are.
We will leave our listeners with that. Blair!
Yes.
Tell us about beavers.
Sorry, I'm looking at this CDC site. It's scary. Okay, anyway.
Yeah, we should link to that if I haven't already said it in the show notes. That should be linked to there.
Anyway, moving on. Beavers, guys, beavers.
Let's talk about beavers. What do you want to tell us?
Beavers may not only help amphibians, but humans who are threatened by climate change. This is a study from Washington State University, and they did some research in Griffith-Pinchot National Forest of the Cascade Range, where they identified, very cool. They identified 49 study sites. They all either had or did not have beaver dams. And they found that the beaver dam sites were 2.7 times higher in amphibian species richness than undammed sites. And when they looked at the types of amphibians, in particular, they saw ones that develop more slowly and need still water that is safe to grow in, which makes sense why the dam would help. And so a couple of species they saw in particular were red-legged frogs and northwestern salamanders. And they are particularly at risk from climate change. So they in particular could really do with that extra help from the beavers. Beavers, of course, were once abundant in the Pacific Northwest, but they were hunted nearly to extinction for their sweet, sweet fur. But in an effort to improve wildlife habitat and mitigate the effects of climate extremes, some land managers have started relocating beavers into the historic locations where beavers used to be. Beavers' numbers are slowly recovering, which is also, as we mentioned, benefiting amphibians. But the important thing here that is not talked about in this particular article is that amphibians are indicator species. That means they're like the canary in the coal mine to water. So when amphibians disappear, that is bad news for everyone, including us. And that is why there's this question about mitigating impacts of climate change. It can mitigate flood problems. It can mitigate water scarcities. It can stop all of these problems or slow them if the amphibians are seeing benefits. So it's basically they're the first domino to fall when you're looking at water quality and water abundance. Mm-hmm. So as go the amphibians, so do we, just later. So that's why if beavers are helping amphibians combat the extra pressures from climate change, that means that can do a lot potentially to help us in the future. So basically a healthy waterway means healthy ecosystems, means healthy humans in the long run, which is why I think it's so important to recognize that when an animal has huge ecosystem services, like beavers making dams, that is more than just saving frogs. And so this proves that they can be reintroduced in these historical ranges and still do a lot of good.
I think that's great. Yeah. I mean, the idea of reintroducing them to places where they have been pushed out, hunted to extinction it could help waterways. I mean, we just had the story this last week of runoff from car tires being toxic to salmon. And, you start adding these things together. How can we mitigate those things? How can we use the natural environment to help itself too?
Yeah, and the really cool thing about beavers too, on a personal note for me, is that in the Bay Area, in the San Francisco Bay Area, I didn't know this growing up, but this is a historical beaver range as well. I always thought beavers were like found in Alaska or something, like totally exotic and different. But in my lifetime, beavers have come back. So they're in the pretty deep East Bay in the Bay Delta, but they are starting to come closer and closer into the Bay watershed. So they are recovering in the San Francisco Bay Area, which is very cool.
That's really neat.
Yeah.
That's super cool. Beavers. More beavers on the show.
That sounds great. There was, wasn't there a reintroduction program in Montana that we talked about some years ago?
Yeah.
But yeah, they recreated just a healthier biome, and it spread out from there with all these ripple effects that, there are more amphibians, maybe the birds are eating better, they're transferring seeds more around because they're eating better, they're, healthier and more birds, and then
they're also just slowing water down, so the nutrient load in the river is better because it's not washing all the nutrients away. So yeah, it has lots of effects.
Flood mitigation and all the rest of it.
Yeah.
Go beavers.
So many. Well, nature is doing its own thing, and it would be probably doing really great if we weren't impacting it. But 2 studies are out this week having to do with humans and nature. And I just can't find anything positive to say about these studies. I'd really like to. One study in particular is out of the Proceedings of the National Academy of Sciences. This was 15 leading international experts based on the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. It was a global assessment, and they discussed the risk to humans' well-being and prosperity resulting from continued degradation of the environment. They were looking at the ways in which nature provides benefits like material goods, food, wood, medicines, non-material goods, and ecological processes that regulate environmental conditions. So water filtration, beavers being part of the waterways, carbon sequestration, storm protection. And they conclude in their study that the well-being of people is not being helped out by nature as much as it used to. We have declining crop yields, we have lower soil productivity, increased exposure to floods and storms, and this is because of the degradation of coastal ecosystems. But pretty much their take-home message is nature's contribution to people is found to be in decline. Nature, you're just not giving back. What's up? And then the other study is a study looking at how humans are impacting nature by what we build. According to a study out of the Weizmann Institute of Science, Ron Milo, a systems biologist, started looking into how the mass of what we are building in the world compares to the biomass in the world. So the mass of all the living things on the planet. They looked between 1900 to 2017. They pretty much figured out comparatively there's been a change over the last 120 years that in 1900, the mass of human materials was about 3% of Earth's total biomass. Materials that we have made to build and use have doubled every 20 years approximately. This is a paper in Nature. Total biomass declined. And we now have gotten to the point where human-made objects, dams, roads, buildings, exceed Earth's total living biomass. It happened this year, maybe, give or take 6 years. I mean, there are fudge factors, and this is an estimate. If you include water in biomass, then it's going to take until about 2037 for us to overtake the living parts of the Earth with the things that we built. That's only 16 years away. Really. So this year, 17 years from now, I don't know. We are at this point where we are building so much. We are truly in the Anthropocene. We are truly in the age of human impact on the planet. Just for perspective, people.
Yikes.
Yeah.
Well, what are we going to do?
There's not much to say about that.
We're on the path of disassembling, separating the nature that evolved on this planet from existing.
Yeah.
It is depressing. Carol in the chat room, you're right. This is depressing. We should move on. Tell us a fun story. Tell me a fun story.
Let me just tell you there's a quote from Eduardo Brandizio, environmental anthropologist at Indiana University in Bloomington, who says, "It's not that infrastructure per se is bad. It's how we do infrastructure that is the problem. Fingers crossed we can make better plans moving forward. Knowledge is power." It's true. It's true. Okay. Do you have good news, Justin?
Oh, it was up to me.
Cemeteries? What? Burial sites? Okay. Let's okay. Bring it.
Well, I wouldn't say it's good news exactly, but it's not bad news. I mean, it's about a dead child, but it's a child that died 41,000 years ago. So, like, it's been a while, right? It's not too soon to talk about this. Question is, did the Neanderthals really bury their dead? Past evidence has shown that they did. And then the skeptics were skeptical, but then the evidence, again, swang in the direction of, it looks like they buried them. Now researchers in France Museum National d'Histoire Naturelle and the University of the Basque Country in Spain have demonstrated that a Neanderthal child was buried 41,000 years ago. There are studies published in Journal Scientific Reports. So this is a site where 6 Neanderthal skeletons were discovered way back in the beginning of the 20th century, early 1900s, for those who don't do that math. The site derived a 7th in the 1970s, early 1970s, belonging to a child of around 2 years old. Since that, the collection associated with this specimen has remained unexplored and investigated in the archives of the Musée d'Archéologie Nationale in France. Researchers stumbling upon this said, "Hey, we have a Neanderthal here. Let's check it out." They reopened the excavation notebooks from the 70s, reviewed the material, and revealing 47 new human bones that had not been identified in the original excavation, which they believe belonged to the same skeleton. Scientists also carried out a thorough analysis of the bones, state of preservation, studied the proteins, the genetics. They did some carbon dating. They went back to the original site, trying to find more bones. They didn't find more bones, but using the notebooks of the people who'd done the thorough note-taking, even though they didn't identify 47 bones, who did really good note-taking, were able to reconstruct and interpret the spatial distribution of the human remains as they had been found on site. Researchers showed that the skeleton had been buried in a fresh sedimentary layer, or not fresh, but separately disturbed from the soil around it. Layer with the head elevated higher than the pelvis. The bones were relatively close together and had remained in their anatomical positions. This preservation indicating that when the child died, it was buried quickly. This is not scattered bones that were windswept or left out in any. So they did, like I said, the carbon dating, which showed it to be around 41,000 years old in the bones. They got the mitochondrial DNA showing it was a Neanderthal, and proteins that confirmed human as well. This is one of the most recently directly dated Neanderthal remains. The new information proves that the body of this 2-year-old Neanderthal child was purposely deposited in a pit dug in sedimentary layer around 41,000 years ago, soon after it had died.
This is yet another piece of evidence that Neanderthals had much more complex society than we historically considered them to have.
Yes, and it's also because we consider that to be a very human trait, burying our dead, caring for our dead, these sorts of things as being very modern, current modern human at least. We don't want to share that with any other animal, let alone with other humans that we, at one point at least, and have still like slowly been having to discard all of these myths of the unintelligent cousin. It seems like they were just as smart as we were. But it's also sort of interesting that when you look at Homo naledi, which is a much older, very different human, very more sort of apish or in between missing linkish. And what we know of them is from a cemetery where they didn't bury their dead, but all the dead seemed to get put, I guess it'd be buried. If you put something way deep down in an intricate cave and leave it there, I think that's on par with burying the dead. Yeah, you know what? Gary and this says maybe humans buried them. So that's actually, it is in a timeline when there would have been some overlapping starting to take place too. So you're right. You want an older example to show that Neanderthals did it first, I suppose. But definitely a Neanderthal child.
Moving away from burial sites and our ancestors or cousins and how they buried their family and their friends, let's talk about bees, how they protect themselves.
I totally thought you were about to do the COVID update right there. And I was like, well, I don't know, this segment, segues.
No, we're going to talk about protection.
Good.
Bees. We know about certain tactics that honeybees have to protect their hives. Blair, you've talked before about the heating process that they do, where a bunch of honeybees will start vibrating and flapping their wings very quickly to heat up the inside of their hive to make it too hot for wasp invaders and other parasites or predator species that come in to attack the nest, the hive, make it unsustainable for them to stay and even possibly kill them. Well, there is a new technique that has been discovered in honeybees in Vietnam. Honeybees in Vietnam have to contend with the murder hornet, the Asian hornet Vespasauror. We've decided to call it the murder hornet here in North America. It has been unfortunately introduced into Canada and has been making its way, trying to spread into the rest of North America. We keep trying to catch those hives and hold off the influx because our honeybees are really not prepared for this hive predator.
They're homebodies.
They're homebodies, yeah. Anyway, these honeybees in Vietnam have been dealing with murder hornets for a very long time. And a researcher from the University of Guelph was looking at some of these honeybees and determined, talking to Vietnamese honeybee farmers and farmers in the area who had these hives, talking about what their bees were doing, what they had seen them been doing, how they were dealing with the murder hornets, because we need to learn tactics for other honeybee species and for potentially these honeybees that we have in the United States. New discovery, Vietnamese honeybees take buffalo poo and they smear it in front of the doorway to their hive.
I'm listening.
Yes. So they took pictures and they have found that these honeybees will go out. They put piles of dung around to see if the bees would come and pick them up. And yes, indeed, they found lots of honeybees. They really liked the stinkier the poo pile, the more they liked it. And they have pictures of the bees going and getting the poo from the dung piles, bringing it back to the nest, to the hive, and placing the poo and shaping it and forming it into little artistic welcoming mounds smeared on the front of the hive. And it keeps the murder hornets at bay. Is it an odor thing? Is it because the smell of the dung overpowers the scent of the honeybees?
Yeah,
is it
like a masking, right?
Yeah, they also looked, it is some kind of a masking. They also looked and found that hives that did not have dung smeared at the doorway were much more likely to be invaded by murder hornet. And as the murder hornets make their way into North America, our honeybees are completely unprepared. Can we smear dung at the doorways of the hives? Can we train the honeybees to do this and protect themselves? Will the honeybees learn in the Americas to use the poo?
Probably not.
For protection?
Yeah, I think the American honeybee is the hybrid poodle of the species. I would take it that any wild form would probably exhibit greater intelligence, greater health, greater anything, flexibility. I don't know. I don't know if that would be, but it's definitely, if buffalo poo works, there's, a lot of these bees are raised on farms. Maybe there's cattle.
Cow pies.
Nearby. You could just put a shovel out in front of the hive somewhere, and maybe that would be fine. Just remember to not step in it next time you go to check on your bees. But
and this is the important question is like, does goat poo work, or is it only softer?
They don't
poo?
know. They don't know yet. They suspect it's an odorant repellent, but they don't know what aspect it is. Is there some aspect of buffalo dung that works particularly well?
If it's pungent enough, I would suppose. I would think it was just sort of masking the molecules that the wasps are looking for. Maybe emanating from the hive?
Yeah. There was an aspect. The researchers took a chemical pheromone that is applied by the hornets to the hive. They mark the hives as a target with their pheromones. And when the pheromone was applied to the entrance of the hives, that's when the honeybees started to apply the dung. So maybe it simply masks the scent of the murder hornets.
Because murder hornets don't have the little GPS dance that bees can do to communicate.
No, they do
have
not.
To actually put a marker out there somewhere.
So, very interesting. Is this tool use by bees? I don't know. I just know it's pretty smart.
Yeah.
I mean, I'm the one who's always saying everything is tool use, so I'm not the one to ask. But it sounds like tool use to me.
Sounds like tool use. Let's say bees use tools now. It's awesome. Okay.
And then the question is, how bad is the smell of these pheromones if they're like, we're going to need something to cover it up. Something strong. Buffalo poo? Yeah, anything to get that pheromone stink out of here. It's really
just
that
wipe
bad.
it out. Use the buffalo poo. Oh, my goodness. This is This Week in Science. If you just tuned in, you're listening to This Week in Science or watching This Week in Science, watching or listening. One of the two. You can do both. If you are interested in a TWIS shirt or mug or other item of TWIS merchandise, head over to TWIS.org. Click on the Zazzle store link, and you'll find our store where we have all sorts of wonderful items with the TWIS logo and also with art by Blair from previous TWIS calendars. Also, there's a link to buy calendars. Supplies are limited. Make sure you get your order in if you have not yet. Get them before they're gone. Get your calendar today. All right, we're going to come on back right now. Time for the COVID update. Time for the COVID update. That's Blair, if you're like me, you're getting tired of this thing.
Yeah.
Right? Are we all just tired?
So tired.
Of this.
So very tired.
Why is it still a pandemic? Why do I have to keep doing all the things? Well, we do have to keep doing all the things to continue to protect ourselves, our neighbors, our loved ones. It is part of the process. However, some researchers at Georgia Tech decided that they wanted to take a look at the relationship, the oscillations between this psychological and physical fatigue that is wearing on people from the constant vigilance that we all have to do with the actual oscillations of the virus itself in our population. So what happens as we're moving through? Basically, what they published in the Proceedings of the National Academy of Sciences is that if people are aware of the severity of the epidemic, they might change their behavior. And if they change their behavior, there's going to be fewer severe outcomes. And so that awareness is important. But short-term awareness is basically what we're good at. Long-term vigilance is very hard, and this fatigue sets in, and the virus comes back again. So there are plateaus and oscillations that are balanced in between cautious behavior and then relaxation of that behavior. The main take-home message is that it's really important to understand that our behavior drives epidemic outcomes. It's not just the data on how many people are infected or dying or, or in hospital beds in ICUs. It's not just that data that's important. It's our actions that are important because we are part of the dynamic system that is this pandemic. So please remember to be vigilant, to continue to wear your masks, to social distance, to do what you can, because if you do what you can today, it's going to reduce impacts on the pandemic later. Right?
Yeah, I mean, it's very easy to be like, oh man, here we go again, without taking any personal responsibility, which I understand. It's not fun to take personal responsibility. It's not fun. I get it. But also, clearly, a lot of people traveled for Thanksgiving.
Yep.
And we're not done with the spike from that event.
No, and because of the delay in symptoms, in hospitalizations, it's going to be with us for quite a while. And we are from Thanksgiving, and then you add Christmas to it, we're going to be seeing the effects of it in our hospitals into 2021. It's not like, oh, there was this, and then people are going to get sick, and then it'll all be better before the end of the year. It's going. Our ICUs are going to be impacted for a while to come.
Right. A million people have said it at this point, but I'm just going to say it again. Yes, your holidays might be sad and lonely this year, but that might be the decision that you have to make so that you can have a holiday season with your loved ones next year.
Long-term thinking. Now, in terms of this long-term thinking, mice, we cure them of everything in all the research that we do. We cure them of depression. We cure them of things they didn't even have. Well, this is one of those things they didn't even have. Mice cannot get COVID-19. The spike protein does not recognize the mouse version of the ACE2 receptor.
This is directly related to my COVID story. I can't wait to hear.
Awesome. Okay. Yes. So it doesn't recognize the receptor. And so, of course, researchers were like, well, we have to cure mice, so we have to give mice COVID-19.
So buckos.
So scientists at UCLA genetically modified mice so that they have human ACE2 receptors, and then they injected them with SARS-CoV-2, and they got sick. They're looking at the effects of that sickness in the animals. They saw lost weight. They saw increases in immune cells, swelling of heart tissue, wasting away of the spleen. This is all very, very similar to what we see in humans who have COVID-19. Additionally, they looked at genes that got turned on and off in the mice that were infected, and they saw that energy generating processes had gotten turned down or turned off in the heart, kidney, spleen, and lungs. And the researchers say if a virus snuffs out the energy generating pathways in multiple organs of the body, that's going to totally wreak havoc. And it does. Mm-hmm. The study revealed also that in these mice that were given human ACE2 receptors and got COVID-19, they found the virus made epigenetic changes to the structure of the DNA in the cells in those organs. And the epigenetic changes may explain the long-lasting effects of COVID-19. Because if your epigenome is getting changed, that's a methylation or a twist or a turn in your DNA that shuts off certain genes from getting translated or turns them down, makes it harder for things to get turned into proteins that normally would get turned into proteins. And while this is not a study in humans, this can potentially tell us a lot about how things are working in the human systems. Anyway, for once, we actually gave mice something instead of curing them of it, but it might help. Blair, what was your COVID story?
So, yeah, speaking of how mice can't get COVID, this is a study out of Stanford University, so it's a bit north of where your study was taking place. They looked at specifically why some animals are more susceptible to COVID-19 than others, which we've reported on a fair amount, saying, okay, looks like cats can get it. Oh, maybe a tiger got it. Oh, looks like maybe pigs can get it. Nope, pigs can't get it. Minks can get it. Ferrets can get it. Yes. So it's been kind of this constant discovery over the past year or so, trying to figure out who can get COVID-19 in the animal kingdom. And so right now where it stands, we know cattle, cats, weasels can get COVID-19, but others, at this point, pigs, chickens, for example, cannot. There was a report, as I mentioned, of an infection in some tigers at a zoo, but that's still, no scientific papers on that yet. But as far as researchers can tell, it's all about the spike proteins to the ACE2 receptor on the surface of the animal cell. So in this study, they used computers to simulate the protein's 3D structures and investigate how the spike protein interacts with the different animals' ACE2 receptors. So basically, they just figured out which lock the key fit in and tried across all these different models. Certain animals' ACE2 lock fits the key better, and that these animals, including humans, are susceptible. And when they looked through their analysis, they also suggest that other species are immune because their ACE2 lack these features. So the key does not fit in the lock. So it's a weaker interaction with spike proteins. All that's pretty consistent with what we thought so far. But this model is really what's interesting because it could aid development of antiviral strategies that uses artificial locks to trap the virus. So that would be one way to prevent the virus from infecting animal cells, have it lock to the wrong thing and prevent it from interacting with human receptors. This could also, this is the thing that I thought was very interesting, help improve models to monitor animal hosts where a virus could potentially jump to humans. So we could prevent future outbreaks by recognizing when there are these locks, these receptors that have similarities between humans and animals. If we know there is a growing infection in an animal species, you can look at the receptor that is being attacked, see if it is similar to a human receptor, and batten down the hatches accordingly. So even though overall it's not a whole lot of new information here, but I think really the technology is the thing that could be helpful in preventing future pandemics, or at least lessening how bad they get.
Yeah, so if we already know in advance that next wave of a virus or something that comes along, if we can see what it's attacking, compare it to our animal models, we can say, okay, it turns out you need to keep your goldfish away from this, because that becomes, I mean, that wouldn't probably be an airborne, but still, whatever it is, we could see what matches up ahead of time. Then, like we do in agriculture, as it is, if there's an outbreak of some, foot and mouth thing, or foot and hoof and mouth disease or something that happens, you have quarantining that takes place around where cattle are. You have quarantining precautions, or if there's an outbreak of something, animal shelters do this a lot. They'll have you step in bleach. I can't even remember what it is that they're trying to keep from getting in or out or spreading.
It's usually just a lot of bacteria that is through fecal matter. Yeah.
But there's always, you can always put that little shield of precautionary steps around something if you know there's a problem there. And if you know in advance, if you know before there's been a huge economic or transmission problem, that all the better. That's fantastic. I love predictive science. I love the stuff that you can do before it happens. It's almost like
Yeah.
preparing in advance for a pandemic and having.
Woo. Let's do that. Yes. So there are a couple of questions that I wanted to answer, to talk about very quickly before we move into the next section of the show. Carol Ann is wondering if any of this is going to help a vaccine for Nipah virus as well. Fada is wondering, can we remove all ACE2 receptors from humans? And then Gaurav Sharma is asking, can CRISPR modify our spike protein to be unlockable by any key? Now, CRISPR is, we don't have a spike protein. We have a receptor. So we have the ACE2 receptor in our cells. The spike protein is on the virus. I don't want to modify that spike protein. I just want to lock it up. So just recognize it and have some kind of molecular lock that goes, nope, you're not going to recognize anything and you're not getting into my cells.
Yeah, so that's basically what your antibodies do, is they give this virus a big old hug. They're like, you're not going anywhere.
Yeah. And then the question of whether we would want to CRISPR our ACE2 receptor, or, as Fada said, remove all of our ACE2 receptors. No,
we need those.
We need those. They're very, very important. Don't mess with the ACE2 receptors. They are a critical part of a regulatory processes throughout the body. It's the angiotensin converting enzyme 2, renin-angiotensin system, and it is involved in water balance, in blood pressure, in inflammation. It is also involved in wound healing, and it is a very essential part of metabolism and your physiology. So let's not CRISPR it, and let's not get rid of it. And then along the line of whether or not any of this is going to help for vaccines for Nipah virus or any other virus, yes. All of the stuff that we are learning for this SARS-CoV-2 virus is going to be information that we can apply to future viruses or to other viruses that we are already working on treatments and vaccines for. So, and it's but different viruses work differently. There is a report out this week for a clinical trial of a universal flu vaccine that had very positive results. It's very exciting. And in the flu vaccine, the flu virus, there's the hemagglutinin and the neuraminidase, 2 parts. That's the H and the N. So you have like H1N1, H1N5, all of these conformations that are the flu virus that we're dealing with. The hemagglutinin part is the head of the structure of the molecule that our body responds to. And it's the head part of it that very often is changing and becoming different enough that our body doesn't know what to respond to. However, we have seen that there is a stalk that hemagglutinin head is on. It's got a neck portion, and that neck portion is much more conserved. It doesn't mutate as often. And people who naturally produce antibodies to the stalk as opposed to the hemagglutinin head can have protection against multiple flu viruses. So that's what the universal flu vaccine that they're trying to create now is doing. It's targeting that stalk that supports the hemagglutinin head. There are issues related to it, but this strategy is one of targeting these particular molecular segments that don't mutate, that don't change very much, that are potentially going to get us where we need to go.
Yeah, and I think also, hopefully, we have learned our lesson, not just in technology, but in importance of addressing things. I think that you will see financing for vaccines going forward and research in these areas to be at a higher level. I think this is going to be really good for the science community.
Yeah,
so I think the technology going forward, we're learning a lot about how to address these things, but I think we're going to learn a lot as a global society to take these threats more seriously going forward. Even though we're
I
not still
do
now.
hope so.
I don't know. Maybe I'm wrong.
But we need to be prepared. And it is time for us to prepare for the rest of our show. We got some good news. We got more poo coming up in just a moment. Thank you for watching TWIS. Thank you for listening. Thank you for being a part of our audience. If you think this is great, share it with a friend. Tell someone about TWIS today. All right, we're coming on back now, and you know what time it is. It's time for Blair's Panda Poo Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You wanna hear about animals? She's your girl. Except for giant pandas and squirrels. There's animal corner.
What you got, Blair?
Well, I'm saving the panda poo for last. You're just going to have to wait. But first, I have a story about dog vocabularies. This is from TWIS Laurent University in Hungary. And they put electroencephalography (EEG) machines on awake dogs. These are not trained dogs. These are not research dogs. These are just family dogs. And they wanted to see if dogs could distinguish specific sounds in human speech, or if they were kind of glossing over specific things when it came to things that sounded like a particular word that they were familiar with. So the first step was to invite dogs to the lab, and they let the dogs get familiar with the room and the experimenters. When the experimenters asked the owner to sit down on a mattress with their dog and hopefully get the dog to relax. Good luck. Then the experimenters put electrodes on the dog's head, fixed it with tape. So far, sounds really relaxing to an average dog. And then with the electrodes on, dogs listened to tape-recorded instructions of words they knew. For example, sit. They also listened to similar nonsense words like sut, and to a very different nonsense word like bep, which I'm going to adopt bep into my vocabulary. I think it's an excellent word. I'm not sure what it means, but I'm going to figure it out. Anyway.
It's what you say when you don't want to fully say burp. It's a little bep.
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 Well, what they found was that most of the dogs learn a few words throughout their lives. Even if they live in a human family and are surrounded by human speech. I have lots of thoughts already about this, but anyway, we'll continue. Their hypothesis was that despite dogs' human-like auditory capabilities for analyzing speech, dogs might be less ready to attend to specific differences between speech sounds. So they might, for example, think sit and sut are the same word. So based on this EEG, they looked at brain activity, they looked at muscle movements, they wanted to make sure they were relaxed. They also wanted to make sure that they were only measuring brain stuff related to what they were listening to, basically. So that's why they were looking at the muscle movements. Some of the dogs who came to the experiment couldn't settle down. No surprises there. I can tell you right now, my dog could not be like, okay, sure, I'll come to this brand new room full of brand new people, let you tape things to my head and just sit. Sure. No, that would not happen. So anyway, some of the dogs couldn't settle. They didn't get to do measurements. But here's something that's interesting. The dropout rate was similar to the dropout rate for EEG studies with human infants. So
that's funny.
Yeah. So far, looking good. The dog brains clearly and quickly discriminated the known words from the very different noises like sit and bep from about 200 milliseconds after the beginning of the words. So that actually is in line with similar studies on humans, which show that their brain responds differently to meaningful and nonsense words within a few hundred milliseconds. So that's, so far, dogs are doing really good. But the dog's brains made no difference at all between known words and those nonsense words that differed in a single speech sound only. So sit and sut to these dogs seem to be the same. And the pattern that they found in the EEGs were similar to human infants around 14 months old. So next time you're mad at your dog, Just keep in mind, they're like a 14-month-old infant with their capabilities in their brain. So the researchers speculate that the similarity of dogs' brain activity for instruction words they know and for similar nonsense words reflects not perceptual constraints, but attention and processing biases. So dogs might not attend to all details of speech sound when they listen to words, but that they could find with further research whether this could be a reason that incapacitates dogs from acquiring a more sizable vocabulary. So a few things. One is, when testing household dogs, there is a huge variability in the amount of training that any particular household does with their dogs. There are border collies that know 200 words. There are household dogs that can't even sit. And that is not the dog usually. I mean, the border collie thing, yes, that is extra special to that breed. It seems a lot of the dogs that can tell, that have the largest vocabularies are border collies. So there's something going on there for sure. But you can tell that within even specific breeds, depending how much time you put into training a dog, they can have a much larger vocabulary and pay way better attention to their human and potential human speech based on that. So there's a lot of confounding variables here related to the home environment that this dog has and the prior training they have had.
Yeah, absolutely.
So there's that.
Yeah, I mean, I've met dogs that verbally could be told at Christmas morning. This is a great example. At Christmas morning, there's this dog that they would tell it, okay, it's time, go get your present. And the dog would go under the tree and sniff for the thing that smelled like a squeaky toy and grab that one toy out.
Yes.
And then whatever it was, they would name it. And they could throw that thing. And this is, I think it's just a lab, like a golden lab or something like that.
I believe it, yeah.
They could throw this toy out with the pile of the other toys and ask for it to go get a specific toy by name, and it would go get that specific toy and bring it out. So dogs do have this amazing capacity to pick up words and do association with words in human language. And they pick it up very quick. I found that for some reason I only speak Danish to dogs. Still haven't quite figured out why that is. But when I'm talking to a new dog and I speak Danish to it, after a while, instead of treat, it knows it's getting kieks. Right, and they pick it up really quick. And it's kind of interesting how well they pick up language for the assumption that we have that they don't really use it.
It could also be the sound that you're making and your body language, a combination of the things, because
I think that helps the learning process. Absolutely. I think that absolutely, yeah, you could go blah, 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 and they're like, uh-oh, yeah. I'm sure that it's part of that learning process. But there's a point where you can remain kind of stony-faced and just say the word. Like, if you say walk around a dog, like there's people who have, if you got your audio up and I'm like, want to go for a walk? All their dogs are freaking out.
Can I say it really loud for Sadie?
Sadie doesn't care about that word, actually. She goes, but yeah, I don't feel like that's it. I feel like the one is when I tell her it's time for dinner, she starts barking.
Okay, I'm excited.
Yeah. But yeah, so I think the main thing that if I can try to interpret this a little bit more, I think what they're trying to get at is dogs don't have complex language skills to the extent that, you know, they could understand the nuance of complete sentences. And like, I think that's really what this is about, is that in order to master a language, you need to understand variance on words. And it's way more complex than something that, say, a toddler can understand. So I think that's kind of what this is starting to get at, is like, this is really cool. Dogs seem to have language understanding about at the level of a 14-month-old infant. And so, yes, we don't expect them to get to a 5 or 10-year-old because they're never going to be able to create complex language themselves with our vocabulary. So,
yeah. Right. Not with our vocabulary. They're not going to use our words. They're not an Alex the parrot.
Exactly. Yes. So it's definitely, they're on the spectrum though, right? Like there are some dogs that are really close to Alex the parrot because they have the same recognition of him where they can understand yellow car.
Yes.
But yeah, so I think it's a little more complicated than the story, but I think it's an interesting study in using dogs that are not specially trained, which I think is a great first step in trying to just figure out what the average dog understands. It's very cool. But also really taking a good look at language recognition. So for all of you out there that think you have really well-trained dogs, maybe it's time to start messing around telling them to sut and see what happens. I'm going to do it and I'm going to see what happens. I think it's going to be very interesting.
Or you know what? Like, how much vocabulary have you actually tried to teach your dog?
Yeah.
Like, how much time have you spent? Do you read to your dog at night? You do that with kids. That's how they pick up their vocabulary.
Have you done training?
Maybe
I do talk to her a lot. I do have like full narratives, which is great. I really like having a dog because, you know, with my husband working nights, so he's asleep during the day, and especially now that I'm working from home, I'm just walking around the house by myself, talking myself through my day. And having a dog here means I can tell her about my day, and it's a lot less insane. And so, yes, she gets a constant dialogue for me. But
and much safer than having a cat.
Yes. For sure. That's true.
We just haven't found out about all the diseases that dogs carry that they give to humans.
Yes.
Because they have bigger poos.
Sure. Yes.
This is my transition to your next story.
Uh-huh. I want to tell you about HMR in giant pandas. What's HMR, you ask?
I have no idea.
Oh, I'd love to tell you. It is horse manure rolling. Horse manure rolling by giant pandas. This is something that the Chinese Academy of Sciences have worked with Beijing Zoo to study in a decade-long study looking at HMRs in giant pandas. Why does this have its own acronym, you ask? It's because about 10 years ago, the research team observed a giant panda in the wild, pausing and rolling itself in a large pile of horse manure. Intrigued, they began watching for other observations, and over the past decade, they documented 38 instances of HMR by wild giant pandas, hence needing the acronym. It happens a lot. Was this the same weird panda over and over, you ask? No. It was dozens of them. They were not just rolling in manure. They were working very hard to cover their entire bodies in the feces. And it was only in cold months, specifically when temperatures dropped below 15 degrees Celsius.
And it's specifically horse manure, or is that just
specifically fresh horse manure?
Okay. This is a big question. Yeah. Why would you do that?
Why indeed? So they suggest that the bears are benefiting from the manure somehow related to the cold. So they studied the manure, as you do. Pandas only, as I said, only bothered with fresh manure. They found in this fresh horse manure, or FHM, if I may coin a term, two chemicals, beta-carophylline and carophylline oxide. They are aromatic, they smell, and they dissipate quickly. They applied the chemicals to hay in the panda enclosures in the Beijing Zoo, and they found that the pandas also liked it, but only when it was chilly. Next, they tested it with mice, and they found that it made the mice less averse to cold conditions. So let me remind everyone, bears, they're good at the cold. That's like one of the things about bears. Think about polar bears, right? So I can't help but wonder if part of this is because they're supposed to be eating meat, but they're sitting there eating bamboo. Are they not as good at staying warm because they're not consuming fat? I don't know. Just a question, not studied in this particular research. But this is the crux of it, is that they found these two chemicals. They were aromatic. They dissipated quickly. They gave the pandas and mice, they think, a feeling of warmth similar to Vicks VapoRub because it was aromatic and it dissipated quickly. It doesn't help them stay warm. It does nothing to help them stay warm. It just takes the sting out of the cold air because of this sensation.
How long does the sensation last? I mean, they have to go find fresh manure to roll in on a regular basis.
It cannot be long. I mean, think how does your Vicks VapoRub last? Probably like a half an hour, right?
Maybe.
But that's direct to skin. So I would guess it's actually way less than that.
Oh, pandas.
Guys, you're just bad at everything.
And now you don't even look like pandas because you put so much poo on your fur. You're brown.
You're literally covered in poop. Do you understand?
Yeah.
Just how can I be more of a panda? Let me think.
Why?
I'm going to cover myself in poop. Make myself more of a useless critter. Let me just...
No. I mean, alliteration.
Cover myself in poop. I know what'll make everyone like me.
Poop.
Cover myself in poop.
Manure. Yes.
Anyway.
All right. There's more to that story.
There's gotta be. I can't wait to hear more about the poo-covered pandas.
Perhaps in 2021.
Research update. Pandas roll in poo because
they think it looks
news at
cool.
12.
Yeah.
Yes.
Guess what, pandas? You're wrong.
But our listeners are not wrong to be listening to This Week in Science. Thank you for listening to This Week in Science once again, for bringing us into your ears, into your brains, and hopefully into your thoughts for the rest of the week as you contemplate the stories from this show. Reminder, calendars are available for 2021. Blair's Animal Corner calendars are available now at the TWIS website. Head to twis.org to order yours today. While you're there, you can also click on our Patreon link. Patreon is where it's our community of supporters, people who support our endeavors to bring science and curiosity and wonder to the world, to share science with you and with others. If you appreciate the show and want to help us continue to do what we do, click on that Patreon link and select your level of support. We have now started annual memberships. There is a discount for becoming an annual member if you are interested in just paying once as opposed to monthly. But yes, annual memberships now available at Patreon. We thank you for your support. Really can't do it without you. All right, Justin, what do you have to say for science?
So this is actually not me saying this, but this is a letter, it's called a call to action, marshaling science for society, that was put together by current and past presidents of the American Institute of Biological Sciences. We're going to have a link in the show notes with the actual, them saying all of the words. But it's pretty long. I have an abridged version, but I did want to read their statement because it's reminiscent of a collage of disclaimers on the show. Over the years. But here it goes. We find the assault by politicians and special interest groups on the use of scientific knowledge to guide public policy decision making alarming and dangerous. The marginalization of scientific information and decision making has significant negative effects on our public health and safety, our environmental sustainability, and our general well-being. We need not look further than the disruption and deaths that have resulted in many countries, including the United States, from failing to use scientific evidence in making decisions on how to control COVID-19 pandemic. Many politicians in the United States and around the world have continued to spread misinformation to promote goals they consider desirable. In the face of this problem, all policy should be based on sound science and its application to dealing with any policy of consequence, including those that address the existential threats to civilization. They go on to explain how science works in a very here's what we do kind of a way. They illustrate the failure of politicians to follow or amplify scientific guidance in the face of COVID-19, which leaving the public to be open to suggestion and opinion that they hear, as opposed to the sound knowledge of expertise and research and doctors. Going on, the progress of science over the centuries has led to our deep understanding of natural phenomena. We must find ways to benefit from that understanding as we move into the future. Let us join together. Scientists of the world unite! There it is. Let us join together to insist on acting logically and rationally in a world so plagued by self-centered short-term goals and the false information that they all too often generate. So this is, again, American Institute of Biological Sciences. They get involved in things like debunking bad information or misinformation about science and promoting good science. It's a wonderful organization. You should definitely check it out. Put the full version, link to the full version, as well as the audio. They sort of did an audio collage of past and present presidents of the organization saying those words and more. But I think that's a very important thing. It's something we've talked about a lot, about science, the scientific community putting their voice out there more forcefully, speaking louder. And I hope, again, as much as I think we've learned for a lesson or two or many from this pandemic that we're still at the height of. So we haven't learned the lesson because we're still at the height of it and we still haven't taken action. But hopefully the scientific community goes, oh, okay, this is our job, and we need to be more dedicated to that than to caring what a politician thinks. And it's a political world, so it's difficult, but there needs to be more expertise in policymaking, because we can't do this again. We can't keep doing this. We can't keep following this path. It's a dead ender. And then, talking about using scientific data in policymaking, armored personnel carriers, long-range acoustic devices, LRADs, assault rifles, submachine guns, flashbang grenades, grenade launchers that go with them, sniper rifles with night vision scopes are not military equipment. They are military equipment, but these are just a few the TWIS local police have been equipping themselves with while dressing up like soldiers in tactical gear and flak jackets to go, and helmets and riot gear to go interact with the public. New research shows that the militarization of local law enforcement through all these armors and combat attire and weapons does not reduce crime. There's not even a correlation of crime reduction with militarization of police. Additionally, researchers found that the evidence that the police use to say that they need equipment is full of discrepancies. They're creating their own data, in a sense, to say that they need things, which is absolutely unscientific and unreliable. It says here, scholars rely on accurate data to track and analyze the true effects of police militarization on crime. Policymakers also need accurate data to base their decisions upon. However, to date, we do not have reliable data on how the surplus military equipment transfers to local police and sheriffs through our federal government and is used. This is LSU Department of Political Science Assistant Professor Anna Gunderson, who's the lead author on the paper, which was published in Nature Human Behavior. So much of the militarization was stopped after the police brutality protest in Ferguson in 2014. And then that decision was reversed in 2017. And we saw what we saw over just this last summer. One of the things that they looked at some data releases that were showing how the use of this military equipment within law enforcement. Data was released in 2014. There was another record released in 2018, and they found that the 2018 data didn't even match the 2014 data in terms of going back and saying what these departments had. Researchers then concluded that promoting claims about the efficacy of police militarization, especially crime rates based on research relying on these data releases by the Department of Defense and local police departments, was absolutely and totally unreliable. When they conducted their own analysis using updated data, combining all the data from all these different sources, authors find no evidence that the military equipment transfers from the Department of Defense to local municipalities, police departments, and sheriff's departments. There was no reduction of crime, contrary to what these locals are saying when asking for their toys. Yeah, we need policy making that is based on reality. You know, if you let anyone make a Christmas list and say, okay, but you have to show a need for your Christmas list, they'll probably come up with some reasons why they need it. That's why you need like a third party to actually analyze these things and see if there's something see if there's a there. And we did see, I think, tremendously frightening use of militarized force against peaceful protesters. I mean, it's one thing if you have gotten these things, you're thinking the terrorists are coming, it's Red Dawn, the Russians are going to parachute in from the, all that's going to happen. You want some backup response for it for in the locals. Okay, put that in a vault somewhere. Train with it once a year, whatever. But they're busting all of this out on peaceful protesters. So obviously there's a very big difference between what they said they needed it for and what they used it for. And what they said they needed it for, it didn't do anything. It didn't work. So roll it back. Send it all back.
You don't need a tank to stop petty crime, petty theft. You don't
They got one in Davis. They got one of these armored
vehicles. Yeah.
And they got an MCAT in the little college town, happy, like low crime rate town of Davis, California. And the people, when they found out, like it was hush hush. Like nobody knew the police department just went out and asked for it and they got it. The town made them give it back or give it away. The town, the people were like, no, you're not driving that around our town. No, you absolutely can't have that toy. And that's exactly what it was. It was a toy. It's so that they could play soldier. It's just very frightening.
It is frightening. Yeah. The management of these things, how violence works, how crime works, gun fatalities, all of these things, having the data to make better decisions will be very helpful. Having people in place to use the data to make better decisions will be...
You're pointing out it's glassed over, but the CDC hasn't been allowed to collect data on gun fatalities. Yeah. Police departments don't always report when they have the information about people that they have interacted with that ended lethally. They don't make the same reports there that they do if they arrest somebody. It's less paperwork, in fact.
Yeah. So let's move on to my last couple of stories. I have brains to talk about because we like using our brains. Mm-hmm. I love my brain. And even in the wintertime, I'm using my brain and I'm trying to keep my brain healthy by learning new things, trying new skills, trying to, I exercise. I keep my brain fit, strong. I try to keep it from withering away and shrinking. Like the shrew's brain. I had no idea. According to a study out in the Proceedings of the National Academy of Sciences, there are shrews. The Etruscan shrew is one of them, but also a different group of shrews as well. they lose their brains in the wintertime. Their brains shrink. There is a group of shrews, the red-toothed shrews, and those shrews, their whole body shrinks down. The red-toothed shrews, they grow a little bit after they're born in the summer season, and then in the fall, winter, everything shrinks, and it is a cycle called Donnell's phenomenon. These shrews just shrink away, and then in the spring, they don't hibernate, and then in the spring, everything starts growing back again. They get big and strong. They eat lots of food, and then they mate, and then they die.
Right.
And that's what happened. They just
to be a shrew.
Yes.
No, thank you.
Oh, yes. All right. So researchers wanted in Wisconsin, University of Wisconsin La Crosse, wanted to know whether other shrew species, also had this brain shrinking phenomenon. And they discovered that, yes, indeed, the Etruscan shrew, which is not related in the same shrew group to the red-toothed shrew, it also has a shrinking brain. But it's very specific.
Have we really checked brains shrinkage? Do we check brain size that much in humans?
Human seasonal brain
things.
Yeah. Do humans have seasonal brain shrinkage? Maybe we should look into it.
Well, but they're doing like brain slices and stuff probably to pulling out the brains probably to measure them in these shrews. Mm-hmm. I don't imagine it's an easy experiment to do on humans these days.
No. But they didn't pull these brains out of the shrews. They stuck them in little tiny MRIs.
Oh, good.
Magnetic resonance imaging scanning, and they looked at 10 shrews over a year. Each season, they scanned their brains and looked at brain volume and at thickness in different areas of the brain over time. And they discovered that the brain volume in these Etruscan shrews decreases in the winter, even though they kept the animals under 12-hour light and dark cycle. So 12 hours light, 12 hours are dark. These shrews shouldn't have known that it was winter, but their bodies did, and their brains shrank.
It's because they were counting down to sex and then death, right? Isn't that the whole...
The countdown. I only have a year. I have to follow this pattern. I only have a year. This is it. It's the countdown. They also gave unlimited food to the animals during that time period and had a very consistent temperature. So there was no temperature swing, no other cues that would potentially make the body's metabolism want to conserve energy or resources by shrinking the brain. And it indicated that this whole thing is related to internal cues as opposed to external cues. In a different group, they took away some of their food in the summertime, and then they were able to find out that there was a decrease in brain thickness then as well. So there are cues like food availability that will influence the resources in the body and where they go in these shrews, but they're internal clocks that really drive the shrews and their brain size. They found a layer of the somatosensory cortex decreased in its thickness by 28% in the wintertime and then grew right back the following summer. And the question is now, we know in mice and rats, and we have some evidence that we've talked about on the show over the years about where new cells in the brain come from. So they now are going to start looking at the hippocampus of these shrews to see whether or not the cells originate in the hippocampus when the brains grow back to their thickness, to try and figure out exactly what's going on there.
Yeah. Okay. So then we're talking about if somebody has, I don't know, fallen on a spike that went through their eye and came out the back of their head, but they survived and they're missing a bunch of their brain matter now because of this accident, but the plasticities allowed them to continue going on their lives. Is this potentially a brain regeneration?
Right.
Therapy thing that
How's
can be
it working?
derived from this? Because I don't know of anything that's just been like, I'm going to get rid of a bunch of brain and I'll put a quarter of it back again. It's no big deal. Like regrowing a tail on a lizard or something. Just regrowing brain tissue. I didn't know that was an option.
Right.
Because
And so now if we're looking at it beyond, we've seen it in mice and rats, but to see this seasonal growth in a shrew, that's another mammalian species that gives it some robustness. Yeah.
Yeah, it's in the family tree somewhere. We're distant cousins. But then the thing is too, maybe you've gotten this far in life and you're not happy with the brain that you have. You're like, maybe I want to, roll the dice a little. See what would happen if kind of got rid of a quarter of this brain and let it regrow back. Maybe that brain will work better than the one I've started with.
Yeah, it's a huge question, though, of, what is happening to them in the winter that, is their cognition, is their ability to think, to function, is it impaired when their brain is less thick? When
and
that
you could totally test all this, right? You could test memory and behavior from one summer to the next.
Yeah.
Teach the shrews a bunch of stuff this summer and see if they retain it after the brain is shrunk and grown back again. See if their personalities change in any way. Now I'm really fascinated with how shrews are from year to year. Are they like completely different shrew the next year, or is it just like I can't remember where I put my keys, but that's okay. I don't drive. I'm a shrew.
That's cool. I'm a shrew. I was one shrew in the fall, and I'm a completely new shrew in the spring. Look at me now. Yeah, the shrew takes Manhattan.
Wow.
It's going to be the next Pixar movie. Yeah. So these
doing that much.
Little teeny tiny shrews. There's a lot more going on than maybe we had thought. And then moving on from shrew brains to how we remember where and when things take place. There are 2 studies out this last week. Both of them led by University of Texas Southwestern, and one published in the Proceedings of National Academy of Sciences, another in Science, that are adding to what we understand about space and time and memory in our brain. Time cells are cells in the brain. They've been discovered in rats. They have a pattern of firing that's like a clock, and they fire and they fire and they fire. It's this very regular firing. And this reproducible firing sequence of this activation of the brain is controlled by 5 hertz brain waves that are called theta oscillations. And the process is called precession. And the researchers at UT Southwestern wanted to know whether people also have these time cells. And in their study, they used a memory task that involved time-related information. They measured the brain's hippocampal region in epileptic patients. So these were people with epilepsy who were going in for surgery on their brains. So their brains were going to be opened up anyway. And during that time that it was open, before the surgery actually took place, they had them do some free recall experiments, reading a list of 12 words for a certain amount of time, like 30 seconds, and then doing a quick math problem to distract them from remembering and rehearsing the lists, and then try and do that recall of the lists over again for another 30 seconds. And there's a word that is associated with each segment of time. So the list is within a certain 30 seconds. The next 30 seconds is recall. Another 30 seconds, you're going to have to recall some words. And so it's this timed memory. And lo and behold, time cells in the human hippocampus. We have firing cells that, if they fire more reliably, they actually predict better how well you remember things related to a particular time. And they call that temporal clustering. So just like in rats, people have time cells. And then in science, they also looked at hippocampal cells, looked at place cells. Place cells are cells that fire when you are in a place. And these cells are activated when you go to a new place, and they fire in a particular sequence. And it's been hypothesized that these place cells are what really help you remember spatial locations. In the brain, they're kind of like these, it's like a sweep of a spot. They will fire before an animal, this is being recorded in rats, they fire before an animal will go into a space, and then they also fire as a review of what was just seen in that place. And the researchers determined that there is this forward and backward playing of these cells that alternate back and forth. And the researchers say while the animals are moving forward, their brains were constantly switching between expecting what's going to happen next, place cells that are just learning a location, and then those place cells firing again in the exact same sequence, recalling what just happened. And it happens within a split second, fraction of a second time frames. So your brain, when you're going into new places, potentially, this is in rats, not in humans, but potentially when you're entering and exploring a new place, you can imagine that you have a little neural radar in your brain that's going, boop, 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 boop. What's here? Oh, boop, 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 boop. That's what's there. Boop, 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 boop. What's in this next spot? Boop, 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 boop. Oh, that's what's in that spot. Okay, we're going to play it. We're going to figure it out. Okay, we're going to replay it. We're going to remember it. Looking forward and looking back. And our brain has these cells that are constantly active, telling us where we are and when we are.
Combining both of those stories. Me and a friend play this game where you say a movie, and then the other person has to guess what year it came out. And you go, any movie, any
so
era.
Bad at that.
But it has to be during our lifetime.
No
right?
recall.
We don't go back and try to do the 50s or something. We do our lifetime kind of a movies. Like, and what's funny is the way that I remember when a movie came out is where I saw it and who I was with. And those two things, who I went to the movie. If I went to a movie alone, I guess I don't remember it. Because I remember where I saw the movie and who I watched that movie with. And that's usually enough to narrow down the year or two of when that movie came out. So that's really interesting. Because it's immediately what made it. Like, that's how my brain absolutely works. It remembers things in time based on the room that I was in and the individual next to me.
Yeah, our brain is this amazing association processor. So if you've got these regular time marking cells firing in your brain, and you have the place cells checking to see where you are, and you have other cells that are recognizing people around you, and your brain is taking all that information in and putting it together in these these slices of creating slices of reality, where they're networks of neurons firing together, the time cell, the place cell, the face cell, all firing together to put a memory into your brain where you can go, oh, that was that theater on D Street, and it was this person, and it was this movie in 1992. I had a letter from a listener, but it was a correction. It really is what it was.
Did we get something wrong?
That happens from time to time.
We did get something wrong. It was a very kind letter from Jennifer Herricks, who wrote in to say, hello, TWIS team. On the last episode, I heard you mention at the end that we no longer need to get polio vaccines. However, we do still receive polio vaccines, as polio still exists in the world. We live in the United States, and both of my kids were recently immunized against polio. And that's absolutely true. There is still a polio vaccine. I think you were thinking of smallpox.
I probably was. Yeah.
Which was globally eradicated thanks to
right.
vaccines.
That's exactly what I was thinking of. Yep.
Because of the polio vaccines, we are close to eradicating polio, which is still endemic in Pakistan and Afghanistan. Thanks for the great show on science. Thank you for catching that, Jennifer. Sometimes doing a live show, we say things and we get it wrong when we're using our fallible human memories. Yes, as opposed to things that are written down in front of us. But I appreciate the correction, and it's an important one to make. But the point still being that, at this point in time, we don't worry about smallpox anymore because vaccination made it disappear. And for a large part, we really don't have to worry about polio here in the United States, thanks to vaccination. There are wonderful, and that was, I think, the big point that you were trying to make, Blair, in that comment. Yeah, but accuracy is very important, and so yes, everyone. Polio vaccines. Your children probably have had polio vaccines. If you had children, you've probably had a polio vaccine. You probably have not had a smallpox vaccine. Probably not. Let's have an end to this show. We're going to bring it to the end. We've done it. Thank you for listening. Thank you for joining us. I do hope that you enjoyed the show. Shout outs to Fada for help with social media and show notes. Gord, thank you for manning the chat room. And Identity 4, thank you for recording the show. Thank you to the Burroughs Welcome Fund and also to our Patreon sponsors for their generous support of This Week in Science. Thank you to Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Schneider, Donathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume John Lee, Ali Coffin, Gaurav Sharma, Shu Brew, Sarah Forfar, Darwin Hannon, Donald Lee Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke Paul Ronovich, Bentley, The Translator, Big Nell, Kevin Reardon, Noodles, Jack Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Melizond, Johnny Gridley, Kevin Railsback, Flying Out, Richard Porter, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood, Conrad Michaels is a Russian super spy, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Matt Mountain Sloth, Jim Drapeau, Sarah Chavis, Alex Wilson, John Ratnaswamy, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Ben Rothig, Gary S., Tony Steele, Ulysses Adkins, Brian Condren, Jason Roberts, and Dave Friedel. Thank you for all of your support of TWIS on Patreon. And if you are interested in supporting us on Patreon, please click the Patreon link at twis.org. On next week's show,
we will be back Wednesday, Pacific Time, 8 p.m., broadcasting live from our YouTube and Facebook channels and from twist.org/live. I don't know how we're getting to all those places at once, but we're going to be in all of them.
It's magic. If you want to listen to us as a podcast, you can search for This Week in Science wherever podcasts are found. So figure it out yourself, I guess. If you enjoyed the show, get your friends to subscribe as well.
And if you'd like more information on anything you've heard here today, show notes and links to the stories will be available on our website, www.twis.org. And you can also sign up for a newsletter that apparently we've started.
It'll come. You can contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in that subject line, or your email will be spam filtered all the way into Justin and my quarantine birthday cake.
no!
If you want to tweet at us, you'd have to go to Twitter, where we are at TWIScience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes due in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember...
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's this This week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And this week in science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science. This week